Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome everybody again to another episode of our show, bringing you another fascinating guest today who has helped and continues to help create a better tomorrow for millions of people around the world. We have the honor today of being joined by Dr. Emilio Amini, Chief Executive Officer of the Bill and Melinda Gates Medical Research Institute, which is a nonprofit organization dedicated to the development and the effective use of novel biomedical interventions addressing substantial global health concerns for which investment incentives are limited. And he leads the Institute's R&D of novel products and interventions for these diseases, which ultimately disproportionately impact the world's most vulnerable populations. Before joining the Gates Medical Research Institute, Dr. Amini served as the director of HIV and Tuberculosis program at the Bill and Melinda Gates Foundation, where he led the organization's efforts on accelerating the reduction of the incidence of HIV and tuberculosis in high-burden geographies, ultimately with the goal of achieving sustained epidemic control. Over the course of his career in the bio-pharmaceutical industry, he's led teams involved in the R&D of novel anti-infectives and vaccines. 1983 to 2004, he led research at Merck Research Labs, involved in the development of one of the first highly active anti-retroviral therapies for HIV, and Senior Vice President of Vaccine Research, the successful development of a number of vaccines, including those for human papillomavirus and rotavirus. Later served as the Senior Vice President of Vaccine Development at the International AIDS Vaccine Initiative, and from 2005 to 2015, he was the Senior Vice President of Vaccine R&D at Pfizer, leading the development of a Prevnar 13 vaccine for prevention of pneumococcal diseases. He's been awarded numerous awards over the years, elected Fellow American Academy of Microbiology, International Society for Vaccines, and the College of Physicians here in Philadelphia. He received his Ph.D. in microbiology, genetics, and biochemistry from Weill Cornell University Graduate School of Medical Sciences, and we are truly honored to have him with us today. Dr. Emilio Amini, thank you so much for taking the time out of your schedule today.
Well, Ira, thank you very much. It's a pleasure. It's a pleasure to be with you.
It's great having you with us, Emilio. I'd love to start off just a piece that you wrote a couple of years back on World AIDS Day, which was entitled "The Experiences That Made Me an AIDS Advocate and Keep Me Going 30 Years Later," where you recount sort of the experiences of your youth and education growing up in New York City, leaving to do your graduate work at a time where HIV was emerging, obviously your critical thought leadership in helping to turn AIDS from this death sentence at the time into a livable condition. And then you come forward in time and you're traveling the world nowadays and you sort of see a lot of what looks like what was happening back in the early 1980s. Can you talk a little bit about, in 2022, sort of this unique schism, and whether we're dealing with HIV or some of the other diseases we're going to be talking about on this show, a little bit of how a lot of this informs your current strategies on dealing with these unmet infectious conditions?
Well, Ira, pretty much like everyone else, what one does at a given time in one's life is always, very clearly, reflects, the previous experiences, right, of one's life. And thank you very much for that introduction. So as you can tell, it's been a long time, right, that I've been doing this. So going back to, the early 1980s. So, when I finished off my postdoc, so as you know, I got my PhD from Cornell Medical. But then when I finished off my postdoc, which was a postdoc working on polioviruses, so remember, this was the early 1980s. So this was sort of at the beginning of, molecular biology and all of the tools involved in molecular biology. I worked with Professor Eckhard Wimmer, who was at what is now Stony Brook University out on Long Island, trying to understand, what it is about, what made poliovirus vaccines, right, on a molecular, basis. So I became very much interested in vaccines. And when the opportunity came to go to Merck at the Merck Research Laboratories in 1983, it was really to a large extent to learn more about vaccines. Morris Hilleman, that everyone who's ever touched a vaccine, knows about in considerable detail, was still there at the time, and I felt that this was a good opportunity to actually learn from an individual who had spent his entire life and decades prior to that time working on vaccines. But then, as it turned out, this is when AIDS, first became evident. Unfortunately, it was what was it? It was 1980 when it was first well, in 1980 was when it was first reported. A little bit before then, and then in 1983, the virus HIV, was isolated and described. So, I'd grown up in New York City. I actually, until I went up for my postdoc at Stony Brook, I pretty much lived in the city my entire life, right? So, and the neighborhood I grew up in was a neighborhood in Greenwich Village. So as you can imagine, AIDS hit it pretty hard in the 1980s. Even though I had left to, go out to Long Island at the time, my mother still lived in the city. So I would obviously go, to visit her very frequently, in the city. And even when I went, to Merck here in the Philadelphia area, I kept going back to the city. And you could pretty much see the devastation, right? You could see the devastation of at that time was referred to as an epidemic, but it very quickly did, in fact, also become a pandemic. And, when the opportunity came up at Merck, when the company made the decision that this was not something that it could ignore, but really needed to work on, both from a vaccine perspective, but even more importantly, from an antiviral therapeutic perspective. Remember, this was a time when very little was known about the virus, other than the fact that it was a retrovirus. That was an opportunity that all of us that were there decided we needed to become involved in, And for me, it was very personal, right? Because a lot of people now don't remember, that you go back to that period of time, all of the big hospitals had what they called AIDS wards, where essentially people who had AIDS went to die. I mean, that was basically all it was. I mean, because there was nothing else that could be done. And the need to change that, was critical. I remember people clearly suffering from AIDS, young men, suffering from AIDS. When I'm walking down the street there in New York, hailing cabs to, go to see the physician or go to emergency room. And it was, it's pretty striking and obvious that this was something that needed to be done. So that's what drove me into doing antiviral work. When the company decided it needed to do this. So from about 1985 until, we got to 1996, when we came up, with some of the first of those of what became known as highly active antiretroviral therapies. And, that was the first time I experienced that one of those situations where, you develop something novel that all of a sudden changes the course of an entire epidemic, right? And certainly within this part of the world, the AIDS wards at the big hospitals literally closed from almost 1 month to the next. Because now, AIDS was no longer a death sentence. Now, a lot more work needed to be done in subsequent years, in order to make the therapies, more easier to use and more available. But in the end, that was a very it was a very unique experience, at the time. But then what also happened, of course, was that even though one could see the AIDS wards in cities like New York closing down, the transmission of the infection in other countries around the world was extraordinarily large, and then particularly in sub-Saharan Africa, right? And so that dissonance that occurs, right, between saying, well, look, we did something, but it's only being effective in this one part of the world. How do we get it to be effective, and how do we get it delivered to every other population that needs it? And that's what drove me back into vaccines, because I felt with vaccines it would be a lot easier. But then in the subsequent years, it also drove me to saying, we really do need to understand how to use, these therapeutic interventions or vaccines as they're developed, not only for those parts of the world in which they're developed, but for true global public health. And that's how I became, more and more interested in global public health.
Excellent. Excellent. And you know, at Bill and Melinda Gates Medical Research Institute, you have really four major global public health, substantial global health concerns: malaria, tuberculosis, diarrheal diseases, and then sort of the basket of maternal, newborn, and child health. 2, 3, and 4 we have discussed on the show. We'll get into those in a little bit, but I'd love to start off with malaria because, you know, I go to the site and you have some, you know, staggering figures here. We hear about the 500,000 people every year it kills, 200 million sick, but then there's this other figure. Malaria may have been responsible for half the people dying who've ever lived, which is, you know, another shocking piece of this disease. And the interesting thing, obviously, from an infectious disease perspective, right, COVID has taken up a lot of the public mind and sort of the popular press the last couple of years. But most recently, actually in the last couple of months, there's actually been quite a bit of exciting action on the malaria front. Certain movement in terms of the Mosquirix recombinant protein vaccine, which I know that you at Gates were responsible in funding. Also some new oral anti-malarial single dose options. Can you sort of give us a little landscape of what we're dealing with in 2022 and some of where you're choosing to focus? Because I know Mosquirix is there, it's exciting, but then there's other stuff coming in terms of higher effectiveness vaccines and so forth. What are you focusing on in terms of malaria nowadays?
Well, you know, for all of these, you know, I'll call them the pre-COVID big diseases, right?
Yeah.
So what are they? They're HIV, the malaria, and TB,
Right?
And it's so for all of these, and you know, and the same thing to a certain extent was true, you know, in the early months of COVID. What did we try to do? We tried to, even though it, you know, in retrospect, it may sound a little bit naive, but the focus is still the same, which is you want to eliminate this. I mean, you really want to eliminate these diseases, right? You want to eliminate the diseases, you want to eliminate the underlying, you know, pathogens and infections. Now, of course, we always need to remind ourselves that with the exception of smallpox, right, and maybe one veterinary vaccine, one veterinary, you know, pathogen, this has never really ever been done, right? Because smallpox was unique in that respect. But still, one wants to make a, you know, a really good attempt at doing it. So in the case of malaria, you know, malaria, in spite of the development of all of the interventions that we talked about, you know, malaria elimination is a challenge and will continue to be a challenge, you know, in parts of the world where malaria is transmitted either throughout the year or even, you know, or seasonally, which is, you know, in most cases, the way malaria is transmitted. So, you know, the original concept of, well, let's get rid of, you know, the Anopheles mosquito vector. Okay, that's fine and good, but you know, that has its issues, and it's, you know, it can be controlled and it can be managed, you know, with indoor insecticide spraying. And certainly one of the biggest impacts has been the use of insecticide-containing bed nets, right?
Yeah.
Which, you know, has pretty much protected the young children from acquiring malaria. But and vaccines like, you know, the RTSS, you know, vaccine, the Mosquirix vaccine that you just referenced, you know, are reasonably effective, somewhat effective. It's you know, maybe not to the level that one would like, but still to a level that will have an epidemiological impact in protecting young children in particular, who are the ones that are much more likely to actually suffer from the negative sequelae of a malaria infection, including death. And that can protect them basically, you know, from that. But and then of course the use of mass drug administration, where one goes into areas, you know, prior to the period of seasonal transmission of malaria, and you treat all the young children with malaria prophylactic drugs, right? That works as well. I mean, it works remarkably well. But of course, that's challenging because one needs to go into those populations for mass drug administration several times over a season, at least, right? So as you can imagine, the logistical challenges, particularly during the rainy season when the transmission occurring, is not easy, right? And can be done, and not easy to do. So there are needs for other interventions, right? So there are needs for developing vaccines that are potentially more effective than the Mosquirix vaccine. There's a need to develop a vaccine which can be effective in preventing infection or reinfection of adults in these populations. Because many adults in these populations are persistently infected with malaria and serve as, it is now well understood, serve as the primary source of the plasmodium, the parasite that the mosquito picks up and then transmits to the kids, right? So in addition to protecting the children, if you really want to eliminate malaria, you have to cure the persistently infected adults and then vaccinate them to prevent them from becoming reinfected, right? And having a vaccine that is highly effective and very persistent in its protection in the adult population is currently lacking, right? So that's where a big focus in the field is, including in our institute, is to develop such a vaccine. So that when you put it all together, between protecting the children through either mass drug administration or insecticide-treated bed nets, or the use of the Mosquirix vaccine, and then treating and curing persistently infected adults, and then protecting them with a highly effective, long-lasting vaccine, you put it all together, then the numbers all of a sudden start to work when you start talking about malaria elimination.
Excellent. And moving from malaria to tuberculosis, here we have a leading cause of death nowadays in the top 10 in terms of all diseases, infectious disease. It's the number one, 1.6 million per year. We chatted about tuberculosis on a show a little while ago, and the fact, hey, it was also the 100th anniversary of our vaccine with the Bcg vaccine developed back in the day before Fleming developed penicillin, so it's been around for a while. Some of the anti-infectives, I think the 1940s and 1950s, they were first developed. Where are we headed at the Gates organization with regard to, because I know your focus here is we need a more effective vaccine on this one. 100 years, we need a new one by now. Where are you walking with this one?
Tuberculosis is a big challenge, right?
Yeah.
And I keep reminding everyone that before COVID showed up, tuberculosis was in fact the number one infectious disease cause of mortality on the planet, right? So, you know, whenever I describe these numbers, it oftentimes catches people by surprise. But you know, roughly a quarter of the population of the planet, is persistently infected, latently infected with Mycobacterium tuberculosis. Those are big numbers, right? And every year you get roughly 10 million cases of active tuberculosis, right? Which people who become persistently infected then, you know, ultimately go on. Not everyone does, but there's a certain proportion that do go on and develop active TB, right? And about 10 million cases of active TB, and that's usually been associated with roughly 1.5 million cases of TB-associated deaths a year. These are big numbers. Now, unfortunately, COVID has blown right through those numbers, right? But you know, like I try to, you know, hopefully explain to everybody, is that eventually when COVID settles down into the epidemiological background, whenever that may be, and whenever it is, TB will still be there, right? And TB will once again become the number one killer, infectious disease-associated killer of people on the planet. The challenge with TB is exactly, you know, what I mentioned. Most people are latently infected, right? They become, you know, latently infected with TB. And not all of them will go on. Actually, it's actually only a relatively small proportion that will go on to develop active TB, you know, in their lifetimes. But the numbers are so big, you know, with a quarter of the planet's population being latently infected, you can imagine why you're getting 10 million active TB cases a year. So the numbers are just enormous. So, you know, control of TB from an epidemiological perspective is really hard, right? Because the way you want to control it is to prevent new cycles of infection with mycobacteria. Well, that can be hard, right? When you've got 10 million active cases. And the current supposition is that transmission occurs from individuals who have active TB. Though there's a growing body of evidence that even individuals who don't have clinically, obviously symptomatic TB are also potentially capable of transmitting TB, at least at certain times. So what's critically important for TB control are three things. One, you need better diagnosis, all right, because you need much more rapid diagnosis. Because typically what happens is that as an individual who is latently infected with Mycobacterium tuberculosis, may not even know they're latently infected, will ultimately develop, you know, when they start to develop active TB, will develop clinical symptoms, right? You know, the usual clinical flu-like symptoms, fever, what happened, you just feel lousy. So then you go, you know, you go look to get treated. But then when you go to look to get treated, you also then need to be diagnosed. And not only do you need to be diagnosed, but you also need to determine whether or not you've been infected with completely drug-sensitive MTB or if you've got drug-resistant MTB, because that's obviously going to influence the therapy that you're placed on. So you can imagine that by the time everything is said and done, it can be weeks, if not months, between the time the person becomes capable of transmitting the infection and the time the person is actually diagnosed and put on therapy. Okay? And then when the individual is placed on therapy, usually effective therapy will render the individual, make the individual non-infectious within a couple of weeks at most, pretty fast. But in order to completely affect the cure of TB with the current therapies, it can take as long as 6 months. right? Using multiple therapeutic options. And if you've got drug-resistant TB, therapy of which has gotten better in the last set of years, it still takes number of months in order to affect the cure. And you therefore have a certain proportion of individuals who relapse because it's tough to stay. I mean, it's tough for people to stay on 7 days worth of antibiotic treatment. Imagine staying on 6 months of antibiotic treatment, right? It's hard, right? So what's needed is better and much more rapid diagnosis, preferably, passive, what I call point of care diagnosis, right? Where you're not relying on an individual who feels ill to go seek care, but that every time an individual, makes contact with a healthcare system in a given geography or in a given country, the person is automatically tested for TB. It's worked for HIV, right? For HIV, particularly in areas that HIV turned out to be, present at very high levels. People would get immediately tested when they showed up for any reason, right? When they go to an antenatal clinic for expecting mothers or anyone else, who would get tested. If you're HIV positive, be placed on treatment. We need to have the same kind of diagnostic capability for TB. So that's being actively worked on, not by the Institute, but by many people on the outside to see, can we develop, better, more effective, TB diagnosis that one can embed in a primary healthcare system. And then from a treatment perspective, one needs to develop better treatment options, ones which can affect cure in a much shorter time frame than 6 months, preferably only a couple of months, right? And that would be tolerable and relatively easy to use. So you get passively diagnosed, you've got TB, present in your oral secretions, you've got TB, therefore we're going to treat you, and here 2 months worth of treatment and you're cured. And you're done. So that's the dream. And one of the areas that we're working on at the Institute with a number of collaborators is to look and develop novel therapeutic combinations, right, that can be used so as to achieve a much shorter period of actual cure than 6 months, preferably within a couple of months. So that's one area. And then, of course, you've always had the vaccine, right? And the vaccines fall into 2 categories, right? You have vaccines that are designed that hopefully will establish an immune state that will prevent infection from actually occurring. Right? And then you've got the vaccines that are designed to prevent the development of TB in people who are already latently infected with the organism. Right? If you do the modeling, it's the latter one, the one that prevents TB, that'll have the biggest epidemiological impact and also public health impact related to it. Now, BCG, as you noted, it's one of the earliest vaccines. Last year in 2021, it was actually its 100th year anniversary. Now, that is used predominantly in young children, newborns and neonates, to prevent infection with TB, all right, and also to prevent clinical TB in kids, because young children, particularly neonates and newborns, when they become infected with MTB, the Mycobacterium tuberculosis, will quickly develop actual clinical TB, right? It's not pulmonary TB, it's what's called miliary TB, but it's more of a systemic disease. But it can prevent that, and has been extraordinarily effective in being able to do that. What it cannot do is that it will not prevent pulmonary TB or the acquisition of Mycobacterium tuberculosis in older individuals. Because after the neonatal period, the next highest period of transmission and infection begins at around early adolescence and goes into young adulthood, all right? Okay. And in that population, for BCG that's given, as a baby, there is no impact on prevention of MTB infection in this older population. So one of the things that we're studying is what we call Bcg Revaccination, following, and this is an ongoing clinical study that we're doing in South Africa, following on an earlier observation from several years ago, where you can take adolescents who are not infected with Mycobacterium tuberculosis with the organism, receive BCG as infants, and now you revaccinate them in early adolescence with the hope of establishing an immune state that'll prevent the potential of becoming infected during that early adolescent, early adulthood period, right? So that study is ongoing. We'll see if we can repeat those initial results. That will hopefully lead potentially to a policy change in terms of how BCG is used worldwide. We'll see what happens. The other approach is to study a vaccine that could potentially prevent the development of TB in individuals who are already infected. That can have a significant impact. You can just imagine it if, in fact, you get a vaccine that can, in fact, do that. And so, again, we're working on a vaccine called M72AS01E, which is a vaccine that was licensed to the Institute by GSK, who originally developed it, and we're still working collaboratively with GSK in the vaccine. And to assess it in a phase 3 study. In a earlier phase 2b study that was reported right before the COVID pandemic, caused us all to hesitate on the things that we were doing.
Right.
Showed some, pretty interesting efficacy in the study in these individuals. Up to between 50 to 70% efficacy, depending on how you define the endpoint of TB disease in prevention of TB, with a consistent observation over 3 years subsequent to the vaccination. So we'll see what happens. And we've got a full phase 3 development program, built around that vaccine, which includes not just the clinical studies, but also includes doing the CMC, the actual, production development.
Sure.
To do. And with the adjuvant, again, working with GSK, because part of the vaccine will be supplied by GSK into the future if we successfully develop it.
Got it. Excellent. Moving on to diarrhea. Second leading cause of death among children. Once the leading cause was rotavirus, and our friend who lives right outside here, Paul Offit, and you were instrumental in developing the rotavirus vaccine. A lot of other things cause diarrhea. I think you're focusing next on Shigella. Can you say a few words about that?
Well, if you look at the 4 big causes of diarrhea in low and middle income countries, what are they? You've got typhoid, and and in fact, the foundation, my colleagues at the foundation funded some very nice studies to develop a conjugated typhoid vaccine that can be used in little kids. So as that vaccine gets rolled out, hopefully we'll have an impact there. That's one area. The other one is cholera, right? And cholera vaccines have been around for some time, of course. And it's a matter of they're just literally being used, in appropriate settings. You mentioned rotavirus, so there you go. So that hopefully we have a vaccine there. So that leaves us with Shigella, right?
Okay.
And Shigella is pretty significant. It's highly infectious. There are multiple serotypes of Shigella flexneri, which can cause very severe and damaging diarrhea in young children, obviously resulting in death. There's Shigella sonnei. That's another form of Shigella. So a Shigella vaccine will have to be a multi-component vaccine. What's been known for a long time, going back to the 1990s, is that the immune response, the antibody response to the surface oligosaccharides of the Shigella coat can be potentially protective in preventing the establishment of a virulent Shigella infection. It's a tall order because it doesn't take a lot of Shigella organisms to actually cause what's known as shigellosis, which is the dysentery that's associated with the Shigella infection. But there are multiple approaches, all funded by my colleagues back at the foundation. There is one particular approach that we're working on that, along with, again, our foundation colleagues, to see if we can develop a vaccine that can elicit, very high levels of surface oligosaccharide antibody responses, immune responses to, again, the 3 different predominant Shigella flexneri serotypes and Shigella sonnei. We're very early on in that program. Right now, it's in the process of development. We're hoping to bring something forward in clinical studies within the next couple of years. And some of our colleagues in the field are also working on similar vaccines, so over the next couple of years, I'm hoping that we'll see some at least early indications of potential efficacy in little kids with these kinds of, conjugated oligosaccharide vaccines. And then hopefully one of them, whether it be ours or anybody else's that's undergoing development, will be successfully developed. We'll see where we go in the next few years.
Outstanding.
But it will have an impact, without a doubt. Also, the other problem with Shigella is, and unfortunately, particularly in low and middle income countries, an unfortunate increase in antimicrobial resistance. Right? Yeah. The fluoroquinolones, which are typically used for treating, shigellosis, the organism is becoming very rapidly resistant. Vaccine is very much needed.
Absolutely. Lastly, Emilio, maternal and newborn health, and this is obviously one that has and you have it drawn out quite nicely, a little complex on the website in terms of sort of the ecosystem between mother and infant and everything that can go wrong. Actually, we had from Merck, we had the Merck Foundation people on talking a little bit about this area a few months ago, and in the United States, we do a pretty bad job as well. But talk a little bit about the dry powder lung surfactant and a little bit of this angle and why you've chosen to approach of all the problems that exist in this ecosystem, why this one's exciting to you.
Well, we sort of made a change in that one, but I'll come
okay, tell me where you are going.
We're no longer working on that particular approach, but
okay.
I'll explain what we are working on at the moment.
Sure.
So first of all, it is a big sort of maternal and neonatal child health, as you noted, is a sort of a catch-all, right? There's a lot of considerations under that umbrella, and as far as newborns, particularly newborns in challenging environments, there are issues related to neuroprotection, right? There are issues during early development. There are issues related to lung development, right? Particularly in premature newborns, as we know. And then a very interesting one area we are working on, and that is early neonatal nutrition, and we'll come back to that in a second. So with respect to the lung surfactant issue, that remains a significant issue, as you know, and in this part of the world, it is dealt with now almost routinely, in neonatal intensive care units, where you ensure the use of an appropriate lung surfactant so that the child's lungs can operate and can expand if a child is born prematurely. But the way in which it's done, as you can imagine, in this part of the world, and in hospital and neonatal units in major medical centers, that's not going to work particularly in low income country field usage, right? So one effort that is being heavily funded and heavily focused on by my colleagues back at the foundation is to try to develop a more facile way of delivering lung surfactant, right, to early newborns, premature newborns in I'll call it difficult field conditions. The dry powder lung surfactant is one particular approach. It's not been completely abandoned. It just wasn't yet ready for full development. Let's put it that way. But there are other approaches which use the more traditional liquid lung surfactants, but it's also a matter of developing an effective delivery system, right, which is much easier to use and much simpler to use in areas where you just don't have people or healthcare workers that are necessarily fully trained, right? So it's got to be something to be easy to use and easy to deliver, and obviously at reasonable cost. So all that work is being very heavily funded by my colleagues at the foundation. One area we at the Institute did get involved in, and again, it started with an original set of interventions that were funded by, again, my foundation colleagues, is the use of probiotics in neonatal infants. And it's just bear with me for a couple of minutes. It's a very interesting story.
Sure.
So in most healthy infants, certainly in this part of the world, and I didn't even know this until, believe it or not, I got involved in this field, some months ago. As a result of just the natural environment and the natural mother's environment as well, one of the intestinal tract of these young children is naturally colonized with a organism known as we call it just B. infantis for short, right? So it's a bacterial organism that has the ability to break down proteins in mother's milk, basically break down milk proteins, and therefore the child can absorb it, right? In the absence of gut colonization with that organism, the breakdown of mother's milk proteins does not occur, right? So that the young child does not get the benefit of nutrition from mother's milk as a result of that, right? And in undernourished children in low-income countries, unfortunately, the gut colonization with B. infantis is a rare event. It does not occur. So in a study that was funded by, again, my foundation colleagues, a study that was conducted in Bangladesh, and the data of which became recently available, the supplementation using a B. infantis probiotic, right, of mother's milk, right, when it was fed to the young undernourished children, had a spectacular effect, all right, on improving the undernourished children's weight relative to size, which is one way in which you determine, appropriate development, just simply by the addition of this one probiotic, right? and because you add it to mother's milk, the child feeds on it. The B. infantis then implants in the gut, all right, and now all of a sudden the child becomes capable of breaking down the mother's milk proteins and getting the benefit from it, right? So that's what this one study showed, but that's what's just simply one study. So given that observation and given the extraordinary impact that was seen with that, we are now working with a couple of organizations that naturally make B. infantis as a probiotic that they add to supplemental probiotics that you can buy in drugstores. In order to have them produced in a way that is acceptable from a regulatory perspective, that they're produced consistently, that they're produced at the right titers and what have you. And once we completed that work with them, which is in the not too distant future, then to bring that forward into a much larger study than the one that was originally done in Bangladesh, so that we can get some very good, statistically pretty robust data that shows that supplementation of mother's milk with this B. infantis probiotic is in fact capable, all right, of colonizing the guts, the intestinal tract of newborn children, and subsequently permitting better nutrition, simply because now the child can actually use the proteins in mother's milk. So it's part of this, increasing understanding of how important, one's biotic environment is, right?
Absolutely.
In good health. And here's a really good example of it. And the fascinating thing about it is that, this is an intervention that would be simple to use, right? Easy to use.
Sure.
Not that expensive, and it's relatively straightforward and something that the mother can do. It doesn't require, a clinic setting, and an enormous difference in the early development of the young child. So let's keep our fingers crossed and hope for the best. But this is one of those, sort of, public health early interventions that can have an enormous impact, right? So
absolutely. I love hearing stories like that, especially when it comes to, some of these, I say natural, but sort of the whole microbiome story, and how we're finding it connected to so many important systems. No, that's extremely eye-opening and fascinating, Emilio. I'm really gonna. It's gonna be an interesting one to follow.
It will be an interesting one to follow. No, and I think of all the areas that we're working in, when you look at it, it's the one that, if it all works out, knock on wood, it's the one that's going to have the earliest impact, right, of all the areas that we're working.
Oh yeah.
This is, it's a simple intervention. So we'll see what happens.
Yeah. Emilio, I think in a couple of days, you're going to be giving a keynote along with Stan Plotkin at the New York Academy of Sciences, entitled "The Future of Vaccinology." And obviously, in the COVID-19 vaccine race, we've seen all forms of vaccinology, from the microRNA technologies to things like the Corbevax sort of recombinant work at Baylor. You guys at the foundation funded Novavax and some of those more traditional vaccines. What are you going to be talking about at the conference, and sort of what gets you excited sort of in 2022 looking at the general vaccinology landscape?
Well, it's so the focus of my talk is going to be that, just like any advance, there's nothing particularly special about vaccinology. I mean, nothing happens in vaccinology that is outside of the advances in scientific understanding and the development of novel technology, right? So the first part of my talk is to go back a full 100 years and just take a look at the vaccines that have been successfully developed and place them within the context of what was known at the time they were developed, both scientifically, both from the pathogen's perspective, the immunological perspective, and what was known technologically, right, related to that. And of course, Stan Plotkin, who follows me, is going to do a much better job of doing that than I ever can. But that's the first part of it. But then it transitions to where are we now, right? And what has happened, particularly over the last set of years. So what we've seen is, certainly in the context of the development of COVID vaccines, we've seen a phenomenal usage of vector delivery systems, right? Whether they be mRNA delivery systems, whether they be the adenoviral vector delivery systems, and a few others. So this started going back a few years ago. First of all, certainly if you take a look at the adenovirus vectors, that started way back 20 years ago with HIV, right? And HIV didn't work all that well in HIV simply because of HIV from an immunological perspective, but the vector delivery system worked. And then, of course, the successful use of vector delivery, such as the VSV vectors and the adenovirus vectors for Ebola vaccines, right, which was the first successful use. of that. But then, of course, that then translated to the development of COVID vaccines, and then a very novel vector delivery system, which is the mRNA delivery system, which was right at the right time of its technological development when COVID hit. You know, it sort of all came together in one confluence there, which allowed then the use of that novel technology for a COVID vaccine that also led to an understanding of mRNA technology that can now be applied to many of the other vaccines that are still, pending, to be developed. So that's one very good example of that. But the other thing that, if you look at novel vaccine, technology, and this is really less technology that's inherent in the vaccine itself, but the technology that you use to understand how a vaccine needs to be constructed, is an understanding of the actual interaction between the pathogen and the immune system, right? Mm-hmm. So again, in a very simple way, you take a look at COVID, the work that was done by my friends and colleagues at the VRC, and people like Barney Graham, John Mascola, and that entire group, that showed that, for the spike protein of the coronaviruses, you need to stabilize it. You need to stabilize it in a given way in order to express those antigenic determinants that will give rise to neutralizing antibodies. So that was knowledge that was applied immediately in the development of most of the successful COVID vaccines, right? Mm-hmm. But that only was applied simply because there was work that was done in the field with other coronaviruses, right? And an understanding of the interaction between the structural understanding between the interaction between those coronaviruses and the immune response, particularly the antibody response. RSV is another example of that, right? Respiratory syncytial virus. Again, the work done by Barney Graham and colleagues, back at the VRC, that showed the importance of stabilizing, though it's a lot more difficult with RSV, stabilizing the fusion protein of the RSV in a pre-fusion state, right? And using that as your immunogen, which, for many years, no one ever understood and wound up developing RSV vaccines that never worked, right? But just all of a sudden they work. So now you've got better and better technologies as time goes forward in really understanding the nature of those immune responses, particularly the humoral immune responses and the antibody responses, right? You can isolate, various monoclonal antibodies. You can assess them at a structural level, how given monoclonal antibodies, interact with, the potential immunogens and surface structures of pathogens. You can, from the immune system perspective, actually look at the usage of immunoglobulin genes, for instance, right? Mm-hmm. And you can look at the usage of immunoglobulin genes and really try to understand, what development pathway, for lack of a better term, do I need to drive those gene products, those, to give rise to antibodies that are going to be, very effective. Yeah. HIV, and a lot of the work that was done in HIV, drove a lot of this work originally. And of course, unfortunately for HIV, what's been discovered is that, driving the immune response for an effective HIV response is going to be really hard. Not impossible, but it's still going to take some time to figure it out. Mm-hmm. Or not to be a lot easier for the coronaviruses, right? Related to that. But again, it's where we sit in our technological environment and our understanding of the nature of the immune responses that we use, and even on cellular immune responses, and understanding of the specific determinants that are presented by various MHC molecules, right, in the context of a given infection, and what that can do, and then translating that into an effective vaccine. So we're moving from a period where we develop vaccines more or less empirically, right? Mm-hmm. I mean, the days of live attenuated vaccines, what were they? Those were empirical, right? The effective, I don't know what immune response is going to be effective, so let me use an attenuated version of the pathogen and hope for the best, right? We don't need to do that anymore. We can understand what we're trying to elicit, and so that's what the future, potentially holds, And as you develop these technological systems and this better understanding, this then becomes part of that toolbox, just as we saw with the COVID vaccine response, that hopefully will be available when the next pathogen that drives a pandemic shows up, because it's only a matter of time.
Yeah. That segues into the next question, perfect, Emilio, because when one puts Emilio Amini into PubMed and your several hundred papers come down, we see pneumococcus and HIV and polio, but when we go to the very beginning of it all, back in, I think, 1980, you published a couple papers on something called Venezuelan encephalitis virus, which I believe, if I looked up the right one, this is an equine virus, if I'm not mistaken.
Probably the only person who's ever read that paper. Yes, but at any rate, yes. And,
the reason I bring this up, and we'll talk about pandemics now or future pandemics, keep our fingers, whatever. But we talked about this principle on the show of One Health and the fact that, yes, unfortunately, there's a lot of nasty stuff still out there, and we have to have a pretty good understanding of the human animal environment connection and how it all works together to hopefully, whether it's the bat or the zebra or whatever it may be, that is serving as that conduit between something very scary and us. We got to be there, as you were just saying, and prepare. I know that a couple of days ago, and as your colleagues at the foundation had a program with Novo Nordisk Foundation talking about some antiviral programs for future pandemics. Give me just your top line on sort of One Health and what you're thinking. Obviously, you have a lot on your plate that you think about, but when you think about all the other nasty stuff that's out there, and it has to be Venezuelan encephalitis, but what are you thinking about? What are you thinking about in terms of One Health programs? What we should be setting up, where we should be investing a few $1 billion to prevent the next encephalitis?
It's a tough question. So I mean, look, so what do we have? What we've got is a world right now which has a lot of people in it, right? More people than it's ever had previously in its existence as a planet. So you've got a lot of people. You've got the ability for a lot of and for continuous intermixing of those populations, right? Which much more so than ever in the past. You've got obviously the contact with, I'll call it the natural world, for lack of a better term, which is a lot more extensive than it's ever been, in the past. So the potential movement of pathogens and adaptation of pathogens, into humans from animals, and vice versa for that matter, the probabilities have simply gone up over time. I mean, there's just no other way to put it, right? And they've gone up substantially. The challenge, unless we all want to live in a mountaintop, by ourselves individually, the challenge is to try to take a best guess as what the next thing is that you need to worry about, right? The challenge with that, Ira, is that, your best guess in terms of what's coming is always colored by what has already happened, right? So in a lot of these efforts to try to develop, potential novel pan-antimicrobial agents, pan-antiviral agents, in the case of viruses, or develop, vaccines that can be effective against entire classes of potential pathogens, that's entirely focused on those classes of potential pathogens for which we have a pretty good idea for which the next major human pathogen might come from. Mm-hmm. And it's all, coronaviruses were always on that list, right? They were always on that list. The influenza viruses, are on that list. The paraflu viruses are on that list. The alpha viruses, of which Venezuelan encephalitis virus is a member, is on the list. In fact, when I was working on Venezuelan acquired encephalitis back in the late 1970s, the reason why it was of such interest, and we were, in fact, the lab I was in was heavily funded by the U.S. Army back then.
Okay.
Was in fact because there was a very large ongoing epizootic, right, making its way up from South America through Central America of Venezuelan encephalitis infections, right? The mosquitoes would feed on horses and donkeys, basically, and would spread the infection on other horses, but then it was jumping to humans, and it was jumping consistently to humans, right? So the concern was that you would eventually, it was eventually going to break into this country because it was making its way up through Central America and then into Mexico, and then who's next? Texas is next, right? Then it was going to come there. Now that was eventually taken care of in spite of all of the efforts to develop a vaccine and try to understand what was going on by simply killing all the mosquitoes. This was back in the days when you could just spray the Rio Grande Valley with mosquito insecticide, and that was the end of that. But those days are gone. But but there's an example of, again, that would have been one that could have broken out into a not insubstantial epidemic. Right? We see it with dengue, right? We see it with chikungunya, right? And so that goes on. So trying to develop interventions, whether they be antimicrobial interventions, antiviral interventions, or vaccines, in advance of then being forced to actually having to use them, right? To actually control a novel epidemic, or heaven forbid, another novel pandemic. The hope is that'll put us in a place where, we're not chasing the next pandemic as we just did, right, with COVID. But, unfortunately, humans in general have this tendency, and maybe it's a biologically ingrained protective tendency, that once you get past an experience in one's community and in one's life where things were really bad and things have gotten better, you forget about the really bad stuff, right? And you never prepare for the next really bad thing that can happen, right? So therefore, the objective here is not to forget it, right?
Right.
And to do the work that needs to be done so that you can be ready, whether or not the next pathogen is another coronavirus or an alpha virus or anything else that can come out of the epidemiological background. And yes, it will come from animals. That's basically where they're going to come from.
Yeah.
But we're too connected to them, right? You're too connected to the world of bats. You're too connected to what's going on with the mosquitoes. You're definitely too connected with everything. Yeah, it's going to happen again. Right?
Yep. Yeah.
So
I have a plan for it. Emilio,
plan for it, at the very least, have the technologies available,
yep.
Which happen, as I said, more by luck than anything else with COVID. Have the technologies available that you can quickly apply, if you haven't already applied them, right?
Yep.
To trying to deal with what appeared. Because until it appears, you don't exactly know what's going to appear, right? You can only take a guess.
Right. Emilio, one final question. There's this theme that underlies a lot of the work you've done and a lot of what you're focusing on now. And I come back to sort of this term of combination, right? You helped turn HIV from a death sentence into a livable disease with antiviral combinations. You talked about new TB combinations. The Bacillus infantis is a unique combination of probiotic materials. You have this really cool paper where you did combinations with the pneumococcal vaccine, with Hep B and inactivated poliovirus and so forth. Are there interesting things we can learn from your space of anti-infective vaccinology for other chronic degenerative diseases? Because usually, usually in the sort of the FDA's world of the past, we develop drug A, B, and C, and they're on the market for a while, and then we combine them much later on. There's been some new guidance lately about how one could develop combinations of therapeutic interventions early on. Do you see some interesting moves on this front? Obviously, whether it's
and I'll tell you the one area we already talked about that we're working on, which is novel combinations of TB therapeutic agents, right? That can shorten therapy. So one of the challenges there is that the combinations that we're looking at are made up of compounds and of drugs, some of which are already registered from a regulatory perspective. Right. Most of them are not.
Okay.
So that question is actually very much a top of mind question, which is to work with regulators. Once we come up with a combination that will meet what our target is in terms of the time required for cure, but then we're going to have to sit down with regulators and work out that here what we're going to want to register is the combination, not the individual drugs, right? But the combination.
Yep.
Now, traditionally, one would have to had to demonstrated the individual activity of those drugs, all right, related to that.
Yep.
And we can certainly do that preclinically, but to do that clinically and then to do for each one of those drugs and then to do it in combination, at least to the extent in which regulators have asked for those studies to be done in the past, is would lead to a very long development period, right? So it's a very real issue, and it's one that is going to have to be addressed, particularly for those pathogens like TB, in which you need to have combination drug therapy, right?
Yep.
Single drug is not going to do it. So finding novel, regulatory pathways to deal with that is going to be critical.
Outstanding. A fascinating set of programs, Emilio. I mean, I'm going to be reading you on. I've been reading you on for the last few decades, but I'm glad to see you're where you are now, taking care of these significant unmet medical needs.
I appreciate it, Ira. And if I can just leave you for just 1 30-second last thought here.
Sure, please take the floor.
Going back to the One Health question, that you asked. So when we look at One Health, we typically when that terminology is used, It's the fact that, humans and other species, inhabit the same ecosphere.
Right.
True. But we also, can't forget that humans and humans, live in the same ecosphere. And
Yep.
what do I mean by that? What I mean by that, and you've heard this as well, over the years, and we see it with COVID. We saw it, years ago with HIV. We see it now with TB, and have seen it for decades with TB. The challenge is that when we apply our abilities as a human species and our capabilities and our technologies to address these infectious diseases, they don't in the end get to everyone on the planet who actually needs them, right? And that happens time and time again. The reason why TB still continues to be a problem is that it is predominantly a problem of low and middle income countries, right?
Mm-hmm.
Doesn't have to be that way. That is, or for that matter, a problem of certain communities in our parts of the world. Doesn't have to be that way. We saw it with HIV. We see it with TB, and we're seeing it again with COVID, right? Where again, we've been able to deal with COVID more or less in high-income countries, become a lot more difficult to deal with in lower and middle-income countries, right? So, in addition to figuring out, how to deal with the pathogens of the future, we also need to figure out how to get these interventions to everybody around the planet who will need them at the time of the next pandemic, or at the time right now of the existing epidemics, because short of doing that, yep, we will never be able to eradicate them, right? We will never be able to get rid of them, right? And they'll always keep coming back to the rest of the world, right? So. And we saw it in real time over the last few years, with the coronavirus and its different variants. Right?
Yeah.
So anyway, I appreciate the opportunity, and thanks.
It was great having you, and thank you for that sobering reminder of what we're dealing with here. So, for everybody that is going to be listening to this episode across the podcast networks or watching on the YouTube channel, again, you've been listening to Dr. Emilio Amini, Chief Executive Officer, Bill and Melinda Gates Medical Research Institute. Emilio, thanks again for taking the time out of your schedule to come on the show. Obviously, thank you for everything you've been doing the last few decades and what you're doing now. And as we like to say on this show, thanks for helping to create a better tomorrow through your work. Really fascinating story.
No, thank you, Ira. Very, very kind of you. Thank you.